So now it is my pleasure to welcome Peng Kong Fox, our Science Engagement Director. She's also a mom to our CACNA1A champion, Alex. So Peng Kong, come on up and she'll moderate our first session. Thank you, everybody.
All right, good morning, everybody. It's great to see you all here. Thanks for joining us. So I'm going to keep this short so that we stay on time, but our first session is this CACNA1A 101 session. It's for any of you out there who might still be wondering what CACNA1A is, what a calcium channel is, what is CAV 2.1, what do we mean by a autosomal dominant mutation or disease. Hopefully these questions will be answered. We have two great speakers this morning. We have Jen Pan, who's a wonderful, brilliant scientist from the Broad Institute, who's been working with us these last couple of years, and then Liz Butler, who's head of genetic counseling at GeneDx, one of the biggest genetic testing companies out there. And I'm going to just embarrassingly say I have no idea who's going first. All right, so everyone welcome to the stage Liz Butler.
Good morning, everybody. So I just want to start by acknowledging that I'm filling in this morning for Laina Lusk from CHOP, who many of you know. Laina unfortunately has laryngitis and so couldn't be here to speak this morning, but these are her amazing slides that I'm going to be presenting. So her name is here in acknowledgement of that. Let's see. So CACNA1A is a gene that codes for a protein that's important in our brain and the way that we function. So when it doesn't work properly, as with any situation where a gene doesn't work properly, that means that the CACNA1A protein itself doesn't work properly. And so we're going to spend a lot of time talking about genes, but the reality is while I personally love genes as a genetic counselor, it's proteins that do the actual work. And so what's most important is what impact a gene change has on the protein itself and the ability of the protein to do its job. And when that protein doesn't work properly, that can impact the way that the brain functions, and that's what leads to the symptoms that we see in people with CACNA1A related disorders. So this is taking you back to your basic science classes, and some of this information might be something that you remember from high school or college biology, or maybe from helping your kids through their classes. So our genetic material is found on our chromosomes. We have 2 copies of each chromosome. One comes from our mom and one comes from our dad. And those chromosomes are made up of DNA that's tightly packaged. And DNA is like the instruction book that tells our bodies all of the information that it needs to know in order to be able to function on a daily basis. Each gene is like a chapter in that instruction book. So each gene is responsible for making one protein, or sometimes more than one protein. And that protein is what actually then does the work in the body. So those gene instructions are made up of DNA. And DNA is basically like the alphabet in that instruction. And there are 4 letters in that alphabet: A, T, G, and C. And so this is an example of DNA here at the top. And your body is able to read that DNA instruction, and it makes something called mRNA. Prior to 2020, many of us probably didn't think much about mRNA, but it's not just for COVID vaccines. It's also present in your body, and it is in essence a way of translating that written instruction from the DNA that makes up your gene. It translates it into mRNA. mRNA is kind of like a temporary thing. It doesn't stick around for a long time. But the mRNA is what helps your body translate that genetic instruction into making amino acids, which are like the building blocks of proteins. That gene instruction, so those letters, the A, T, G, and C, that are part of the mRNA, are basically read in a sequence of 3-letter words. So all of your kind of genetic material and everything that helps your body run comes from these short words that are basically these 3 DNA letters, as you see here. And so that mRNA, those first 3 letters, code for one protein. That's that little ball there. That protein is a tyrosine, abbreviated TYR. And then you see the next 3 letters code for the next building block of that protein, which is a serine molecule, and so on through. And this will be important when we start to talk about genetic variants a little bit later. Once that protein is completely assembled and all of those building blocks are there, you have a functional CACNA1A protein, which makes up what's called an ion channel. And so the CACNA1A protein is responsible for regulating the amount of calcium that flows in and out of a cell. It's important for cells to maintain the right amount of calcium. And so having that protein made correctly is really important for helping to regulate the amount of protein that's going in and out of the cell. So this is just sort of showing us, again, to illustrate that gene, so the DNA that's making up that written instruction is your instructions. It's almost kind of like a recipe. The mRNA and those proteins are like the ingredients that go into it, and at the end you make a protein, which is your final product. So in this example would be like your cake. This is an example of a neuron, and you can see here that this is the CACNA1A encodes for these channels, which are what are helping to control the flow of calcium in and out of the cell. And the ion channel itself is really important in the brain. And so these ion channels help to control messages that are sent between the neurons in the brain. The channel works a little bit like a traffic cop. So when calcium enters a cell, it allows sort of these messages to come in and out. And it's really important for these neurons to be able to communicate with themselves properly. And when they don't work and those messages aren't being sent in the proper way, then that's what leads to the symptoms of CACNA1A disorders. Okay, so we're going to think about different types of genetic variants, and I think that the easiest way to illustrate this is by thinking about just a regular sentence that you would think about. And so the example of our original sentence is, the gray cat ran down the hall. One type of genetic variant could be a change to the letters in that sentence that impact the spelling, but they don't impact our understanding. So there's 2 different ways you could spell gray, G-R-A-Y or G-R-E-Y. Either way, we understand what that sentence means. You have variants in your body that are benign, that are just like this. So there's a change. It doesn't match exactly what we would expect, but it doesn't impact the way that your body understands that information. It doesn't impact the way it makes a protein or how that protein works. And those are called benign changes. There also are changes that we consider pathogenic. And so this is a change where you're altering that DNA instruction, so that sentence, in a way that your body can no longer understand the information in the genetic instruction. And so it impacts the way that that protein is made. And an example would be if you misspell gray as G-R-I-Y, you're not really sure what that is. Like, what kind of a typo is that? Was it supposed to be gray, or was it supposed to be something else? Sometimes we can have a change that is a variant of uncertain significance, which you'll often see abbreviated VUS, and might be referred to as a VUS or a VUS. We like to have multiple different names for things in genetics just to make it extra confusing. So a variant of uncertain significance is exactly what it sounds like. It's a change in the DNA, and we, as scientists and healthcare providers, aren't actually sure what that means for the way that your protein is made and your body works. So we don't know if that's a benign change or a pathogenic change. It means we have just kind of limited information. We don't have enough information to assign it to one of those buckets as being benign or pathogenic. Some of you may have had a genetic test report come back in your family with variants of uncertain significance. These are relatively common in genetic testing. So when we find a variant of uncertain significance, there's a number of things we can do to try to figure out if that might be benign or pathogenic. Some of them might include testing other people in the family to figure out if that change was inherited or not. We can look to see if this is present in healthy people in the population, and there can be functional studies, which you'll hear a lot about from some of the researchers later who are doing functional studies to try to better understand the way that genetic variants impact the CACNA1A protein. These work really well for helping us, but sometimes we're still left with variants of uncertain significance even after we've done the most that we're able to do. There are different types of genetic changes. So the one that I showed you before is an example of a change of a single letter. That's what we often call a missense substitution. But there can be other types of changes. So for example, an in-frame change. This means that there is information that's completely missing or extra in the protein. So this would be just like taking out a whole word. As you see here, instead of saying the gray cat ran down the hall, we say the gray ran down the hall, and we're missing the word cat. And so that's an example of an in-frame change. You're removing an entire word. A nonsense change is a situation where we basically stop making the protein, so we only make part of the protein. When we do that, our bodies usually recognize that we don't have a full protein, it's not going to be functional, and so your body just degrades it. So in essence, you don't have as much of the protein as what you would typically expect to have. So an example of that would be just saying the gray, and then there's nothing else after that. You don't have a full sentence. Another type of genetic variant is a frame shift, and so this is where we add or remove a single letter. And remember earlier I showed that when you've taken the DNA, you make the mRNA, it reads those letters in a sequence of 3. So if you remove one of them, you're shifting that whole sequence over. And so in this example, the letter Y has been removed from our sentence, and so you see that that C shifts over to the prior word, and everything else gets shifted over. And suddenly, from that point on where that frame shift happens, you no longer understand anything, so that protein stops making sense. A frame shift has similar consequences to a nonsense variant in that your body generally recognizes, this is nonsense, I don't know what this protein is, I don't know what to do with it, and it degrades it. Another example of a type of variant that we can see is a whole gene deletion, and this is where one of the copies of the gene is completely missing. So you just don't have that gene at all on one of your chromosomes. And so, some of these changes, we've got a complete protein, but it's not made the way we would expect it to be made. And in the bottom 3 examples, we have an incomplete or completely missing sentence, or an incomplete or completely missing protein. Another type of genetic variant would be a splicing variant, and this is where we are either incorporating extra information into the protein that shouldn't be there, or we're missing some information that we're supposed to have. And so in the top example, you see the gray cat, and then we suddenly have this randomly inserted information, the orange cat climbed a tree, and it messes up the whole meaning of that sentence. Below you see the gray, and then we're missing the rest of that sentence, and it goes right to the second sentence, the dog chased the car. In these splicing examples, generally what you end up with is either an in-frame variant, so you're missing information, like a big chunk of the protein, or you end up with one of those frame shifts where you basically move out of that series of 3 letters and suddenly everything in the protein stops making sense. So when we have variants in the CACNA1A protein, one of the things that you're going to hear more about today is whether those are gain of function or loss of function. And so this is showing sort of our normal CACNA1A protein that's functioning the way that it's supposed to, and it's regulating the flow of calcium ions in and out of the cell. A loss of function variant would be one where remember you have two copies, one from your mom and one from your dad. One of those two is not working the way that it would be expected to be working, and so you're making 50% or half of the amount of the CACNA1A protein that you would expect to make. Another type of variant could actually result in gain of function, and so this means that we have two copies of the CACNA1A protein, but one of them is doing something it's not supposed to do. Generally, that means that that channel is letting too much calcium into the neuron. So it's staying open and it's allowing more calcium in. It's not maintaining that balance of calcium the way that it should. So in our prior example, we talked about some of the variant types where we could have an incomplete or missing sentence in the gene. So those nonsense and frame shift variants and those splice variants, they typically lead to loss of function. So you're not making one of those copies of the protein. Generally, then, gain of function variants are those missense changes where you're just changing a single letter in the DNA sequence, and so you're making a protein that is maybe different in one place, but you're not missing the whole thing. There are some differences in what this means in terms of the impact on your body. So people with variants that are predicted to be loss of function are more likely to have ataxia. And this could be consistent or it could be episodic ataxia. In individuals with loss of function variants, they may or may not have seizures, and if they do, they tend to start later. Gain of function variants are more likely to be associated with hemiplegic migraine, severe seizures leading to status epilepticus, focal seizures, and low tone. And so there can be variability here, but you do tend to see what we call genotype-phenotype correlation, which means that the type of variant and the impact on the protein can impact the symptoms that you see in the person. And so you may have heard the term or remember the term autosomal dominant inheritance. So remember that we have two copies of each of our genes, one that we inherit from our mom and one that we inherit from our dad. In families where we see familial CACNA1A disorders, so a parent has it, you can see on the right that there is one copy of the mutated gene. Each of the children has a 50% chance to inherit the normal copy of the gene and a 50% chance to inherit the mutation. So an individual with a CACNA1A disorder who is having children themselves would have a 50% chance of passing on a CACNA1A disorder to each of their children. Many individuals that have the neurodevelopmental CACNA1A disorders have what we call a de novo variant. So this means that both of the parents are healthy. They don't have a CACNA1A disorder themselves. The variant in CACNA1A is new, so it arose in either the sperm cell or the egg cell when it was being made, and so that variant is new in the affected child and is not inherited from one of the parents. When that happens, that means that future children of that parent have a much lower likelihood of having a child with a CACNA1A disorder in the future, and we generally say the chance of that happening again is less than 1%. The reason we don't say it's zero is because every now and then we see something called parental mosaicism. So this means that a parent has normal CACNA1A genes in most of their body, but some of their cells, including the germ cells, which are what make the egg or the sperm cell, have a CACNA1A variant. And so even though the parent themselves does not have a CACNA1A disorder, they have a 50-50 chance each time, up to a 50-50 chance each time, to potentially pass that on. Parental mosaicism is rare. This is not something that happens commonly, but we know that families often are looking for as much information as you can get about recurrence, and we always want to be clear that we can't give 100% guarantee, even with a de novo variant, that there wouldn't be future children impacted, but we know that there's a greater than 99% chance that this is not the situation. But but it's important just to be aware. And I just wanted to wrap up talking a little bit about spinocerebellar ataxia type 6. So CACNA1A is an interesting gene in that it actually codes for 2 different proteins. What we spoke about was predominantly talking about the CACNA1A impact that leads to neurodevelopmental symptoms. And so those are pediatric onset. You generally see stable but consistent developmental issues, ataxia, and may also see things like autism, seizures, migraine, and hypotonia. Spinocerebellar ataxia type 6 has very different symptoms, as you may know. It's adult onset, typically starting in the 40s and 50s, although the range is pretty broad. It's a progressive disorder, which means that the symptoms get worse as a person gets older. And the predominant symptoms are ataxia, and you don't see some of the other developmental features. So the genetics of CACNA1A neurodevelopmental disorders are different from the genetics of SCA type 6, and I think this is important to be aware of even if you or your child have a CACNA1A neurodevelopmental disorder, because if you're looking up information, you'll often find information on SCA type 6, which can be confusing. And if we have any SCA type 6 families in the room, this is sort of the piece that will apply more to you, because the genetics is actually pretty different. It's caused by a repeat expansion, which means that there's this series of CAG letters at the end of the CACNA1A gene that are repeated over and over. And most people have CAG repeated 20 times or less. In people with SCA6, when that gene is being copied, something happens and your body gets confused, and it makes that number of CAG repeats much, much longer than it should. And so you end up with way more, and this causes the protein to basically get stuck and doesn't function the way that it's supposed to. These are almost always inherited. They're generally not de novo, so the genetics works definitely a little bit differently. And there is specialized testing needed to look for those repeat expansions causing SCA6. So the gene sequencing that we often do for genetic testing doesn't work. You have to order a specialized test for this. And this is because it codes for this different type of protein that plays a different role. It's what we call a transcription factor. And it includes part of the CACNA1A gene and sort of this additional area where the repeat is. And so the genetics is just a little bit different in the way that our body processes this other protein. And so with that, I'll wrap up. And do you want to take questions at the end? Yeah, okay, thank you. Thanks, everybody.
Thank you so much, Liz, for that great talk. And yes, I'm going to welcome to the stage Jen Pan from the Broad Institute now.
Good morning. Thank you so much for the opportunity to be able to share with you, to talk to you about some of my research on CACNA1A, and then to explain how do we look at the function, the biophysical function of this protein that Liz just introduced as a molecule that is very critical for brain function. So I'm Jen. I'm an investigator at Broad Institute of MIT and Harvard, and this is our research building, and my office is right on the 6th floor. Before I start on the research part, just as a way of introduction, I wanted to tell you my journey of getting here. I had the honor of meeting with Sanitha and Lisa, your tireless leader at the CACNA1A Foundation in 2021. And I was very much impressed and inspired by their energy. And then afterwards, many parents that come Debbie during the conference or outside of the conference. So I started my research career studying a very closely related gene, CACNA1A, CACNA1B, so it's one letter difference at Brown University. And after that, I went to a couple of different places, and then I start building my lab at Broad Institute of MIT and Harvard at Stanley Center for Psychiatric Research. My main research direction is to identify and validate protein targets rooted in human genetics to understand the disease mechanism and then to find potential therapeutics for psychiatric and neurological disorder. So during the period of time I was 5 years ago, I was involved with this the Epilepsy Center Without Walls Center that organized by L. George at Northwestern. And then in that effort, I was studying a number of different ion channels that includes SN2A and CACNA1G. And Elle and Ingo brought to my attention CACNA1A, and I have since met with the foundation and the parents, and I started to look into how does the genetic mutation alter the function of this protein, and that brought me to here to talk to you about what we know about this protein and how we study them. There is a level of personal and intellectual satisfaction that I start to work on something that is close to when I started my scientific career. More than 20 years ago, I feel privileged to be here to share the research with the families who suffer from CACNA1A disorders. Your courage to lead through these difficulties is our, as researcher, inspiration to work on these mutations to find the mechanism by which CACNA1A cause these disorders, and ultimately to be a partner in your journey to find a cure. So this is a quick outline of what I'm going to talk about. I'll explain to you what is a voltage gate, what are the voltage-gated calcium channels, and where do they reside, where do they express, and how do we study the function of these channel proteins. And I'll talk very briefly of, we characterized a cohort of de novo mutations that Liz just talked about that in the kids but not in the parents. And this is mostly from the developmental cohort published in 2020. I'll try to explain a little bit of the complexity underlying an interpretation of the variant function. There is a clear, sometimes it's very clear, the channel allows more calcium to come in. Sometimes it's very clear the channel to allow the less calcium to go into the cell. But sometimes it's a little in the middle, it's a mixed function. And as Liz alluded to, ultimately, she has the genetic information, and then when it gets to the cellular molecular function, we start to understand what does this genetic code, what does this change in the letter exactly mean at the protein functional level. But that's not the end. How do we get to once you well, first of all, for the UVSs, the variant of unknown function, can we dissect to figure out whether or not they're benign or pathogenic? And then from there, to really understand, once you have a variant, can you predict the clinical outcome? That's the ultimate goal. And how do we care with this information, care for patients with this information? So voltage-gated calcium channels support a diverse array of calcium signaling. Calcium signaling is very essential for regulation of a diverse set of crucial functions in the human body, such as overseeing cell death, gene transcription, muscle contraction, exocytosis, neuronal transmission, cell motility, and cell growth. It's essential. Calcium is essential. There is across the cell membrane, there is a 10,000-fold gradient difference. So extracellularly, the concentration of the calcium is 10,000-fold more than intracellular. So cell has developed a very exquisite system to regulate when does calcium go into the cell and how do they get into the cell. So voltage-gated calcium channels are these large transmembrane proteins sit in the middle of the plasma membrane, and they're gated. they're controlled by the voltage of the membrane. And so many cells in our body, their voltage don't change a lot, but there are excitable cells, such as neurons and muscles. Their voltage change very drastically based on their activity. So these channels, when the voltage change in the plasma membrane, this channel's activity can go up, they can open, and they can close. I'll go into it a little more in the next few slides. But basically, when they open, just by the force of diffusion, there's a lot of calcium coming in, and then calcium will do their wonderful things in the cell. And then when the membrane potential goes to hyperpolarized voltages, these channels are closed and don't let calcium in. So that is, in a nutshell, how these proteins, voltage-gated calcium channels work. And then there are 10 genes encode voltage-gated calcium channels. They're roughly divided into 3 families: CAV1 family, CAV2 family, and CAV3 family. CACNA1A is the first member of the CAV2 family. That's why they're called CAV 2.1, and then sometimes you can see there is alpha 1A, and I'll explain it in the next slide. So these 10 genes, they have very different distributions across different tissues in the body. And then they also have different pharmacology, different small molecule or toxins bind to them with different specificity, selectivity, and potency. They have very different gating properties, which means that how voltage can open them and how they respond to those opening is very different. Kinetics is different, voltage dependence of properties are different. And then they have very different modulatory patterns. Their target protein, downstream target protein is different. So altogether, they regulate many, many different functions in your body. And CACNA1A, as Liz mentioned too, is very critical in the brain function. It plays a major role in neurotransmission release in the axon terminals across CNS, as well as in the neuromuscular junction. It also mediates dendritic calcium spikes and excitability. So in this schematic, the neurons, the cell body are the bigger one, and then neurons communicate by chemical and electrical signals. So they send axons to the next neuron and then talk to them. And then the CAV 2.1 are these critical communicators at the end of the nerve terminal to talk to the next neurons to talk about do I pass this information on or not. So this is the so this is a 2D topology of how this channel is. The CACNA1A gene codes the main subunit of a multi-subunit complex that we call voltage-gated calcium channel. It has auxiliary subunit, which beta subunit is very critical for bringing this alpha 1 subunit to go into the membrane. But this alpha subunit is a business end of the voltage-gated calcium channels, which containing the gating mechanism, how they respond to voltages. It contains the pore, the ionic pore where the calcium can go through, and it also contains interaction with the other auxiliary subunit. And then if you just stretch it out in a 2D space in the membrane, it is composed of the 24 transmembrane segment, and these 24 transmembrane segments form into 4 different domains, and then the domains wrap around, and then the core is in the middle when they wrap around. So this is, in general, how this protein works. And then they also have very different expression pattern. So you may not be familiar of looking at this picture. So it's a heat map. The higher the expression is, the redder the signal. And then we have the, this is the cortex, this is the striatum, this is the cerebellum. So CACNA1A is widely expressed across the brain. but particularly it has very high expression in the cerebellum, Purkinje cells, as well as the hippocampus. And that's why some of the symptoms may be derived from cerebellar-related neurocircuitry. I'll skip this. This is more of a single-cell expression analysis. We were trying to see, even in the same region, what type of cell express more or less of CACNA1A. There's inhibitory neurons, there's excitatory neurons that play different role in the different circuitry. And then this is the technique that we use to study this type of channel. It's called patch clamp technique. This technique was developed by CACNA1A. They won a Nobel Prize for discovering and implementing this technique to study the class and the cellular function, cellular physiology. So what do? We use this glass pipette that has very smooth surface at the pipette end, and then we put them closer to the cell. And then when they touch the cell, they form very tight seals. When they form very tight seal, we can break that little membrane within the contact space, and then the pipette have access, electric access to this cell that we can study the electric properties happening at the membrane of the cell. And so here is a real pipette patching on a neuron, and here is explanation. As I mentioned to you, the channel exists in the most simple way in 3 conformations. They typically, at the resting membrane potential, they're closed. And then when the voltage decrease, so typically cell membrane is around, neuronal membrane is around minus 70 millivolts. And then so when the action potential comes in, they're called depolarized. And when the cell membrane is depolarized, these channels open. And then sometimes when they open, they can go into these inactivated states that they are unable to open again. So there is a period of silence before they can open again. And then once we have this technique, we could apply different voltages externally to the cell membrane and to interrogate the function of these channels. I wanted to be cognizant of my time. 5 minutes? Okay, all right. So I'll go a little faster. So using these techniques that we're able to start to look at some of the channel properties that we call, this is called an IV curve. It stands for current voltage relationship. It's very characteristic for any particular gene product, any particular protein. So they usually open at a particular voltage, and then they peak, and then they go back. And then we derive these curves to understand how they respond to voltage, at what voltage they start to open, at what voltage they open 50%, and then at what voltage they may open completely. And then we also use these protocols to try to get at how they close, what kinetics they close. They close faster, they inactivate faster, and then so on and so forth. So to study a large number of variants, the manual patch that was originally developed is not fast enough for us to understand many variants at the same time. So we have developed this technique to building a very efficient transient expression system to investigate function of CAV2.1 with missense mutations, with many missense mutations at the same time. I'll skip really quickly, but it's a technology that allow you to patch 384 cells at the same time. So we developed this technology, and then we characterized, we can parallelize characterization of many different missense variants of CAV2.1 all together at the same time. So I wanted to show you some of the results that we used, what I'm talking about, these techniques, this automated patch, and then validated with manual patch in a large developmental cohort. So we took the CACNA1A mutation, missense mutation, in this cohort that's published a few years back, and this is the developmental delayed cohort. And there are 36 de novo variants reported in this cohort, and we made all of them. And then to compare with the population, we also looked into 8 nomad missense variants. So these are the missense you can find in the general population. So when we did the experiment I just described to you, we found, first of all, we found many of them in our experimental system do not express enough, do not have enough of the current that we can detect, which we generally interpreted them as a loss function. So for the remainder of the missense variants that we can have detections of their currents, we further analyzed their biophysical properties. So these are the remainder of the variants that we analyzed. I just wanted to give you some flavor of the different kind of variant, what do they do to the channel. So for example, this first one, if you look at the, so some of you know about the A713T mutation. This has been reported and studied before, but it's also in this cohort, so we also studied them in this study. So the A713T shifted the voltage dependence of activation extremely to the left, which means that at smaller depolarizations, they would open. So they would allow more calcium to go into the cell. At the same time, you can see that the shape of the mutation is, the currents are very different. So as a wild type inactivate, these mutations do not inactivate, they persist. So that also allows more calcium to come in. So based on these, we generally interpret this particular one as a gain-of-function mutation. There are many other examples. I won't go into the details of them, but sometimes the two properties can go conflict of each other, and now we call it mixed mutations, which means, for example, you don't have enough of your channel to get to the cell surface, but when you get to the surface, they shift how they sense the voltage. So there are a lot of different phenotypes, but in general, so we try to visualize the impact of missense variants in this type of pentagon plot. In here, that we plotted the property of 5 biophysical properties we interrogated as a pentagon, and then we calculate the z-scores, which means how different is your variant compared with the reference. And if they allow more calcium to come in, it's extended outside. If they allow less of a calcium coming, they're shrinking into middle. So you can see that the domain 1 doesn't seem to impact a lot. Domain 2 has a lot of variance change, particularly the V-half of activation and the current density. And then domain 3 as well, a lot of current density change, which is the north vertex, and as well as this northwest vertex. So we use these different ways to visualize them. And I'm going to skip the next couple of slides, and then just, we're trying to understand what is the physiological impact of these different biophysical properties come together. And we use in silico modulation, simulation to do that. But I'll skip that, and I just wanted to say that, you know, from what Liz was talking about, the genetic mutations onto one gene, what we're trying to do, what we can do with cellular and molecular characterization is to understand exactly what is the impact of that one DNA change onto how this protein responds to voltage. But that's not the end. That's not even the beginning of the end, but hopefully that is the end of the beginning. So once we have this information, for example, with some of the VUSs, variant of unknown significance, we can put them in this type of functional study and understand exactly what those variants do to the molecular function. And then this is very much at the molecular level of this molecule. What does it do in a system that is very artificial? Downstream, there's, what does the mutation do in the neuronal cell, in the different types of neuronal cell, inhibitory neurons and excitatory neurons? What is the function in the neural circuit and animal models and all the way to clinical phenotypes? There's a long list after that. But this is the second step, the first step after the genetic information for us to get hold of what does it mean to have this mutation at the molecular function level. So with that, I wanted to thank all the people who actually did this work back in the lab. Wanted to thank the foundation, the Lisa, Sunitha, Pankaj, the interaction with them just being incredible. It's very inspiring. And all of you who are here, who are parents, who lead through this difficulty of trying to understand what happens and how we can make improvement. Your life experience is our inspiration to do this research. I want to thank Elle and Dennis for getting me involved in this CACNA1A conversation, and Ingo and Laina. And then thank you for your attention.
Liz, do you want to come back on stage, please? I think we've got time for like 10 minutes of questions. All right, so I know that was a lot of information thrown at you, and it's hard to sometimes take these scientific terms and explain them in a way that everyone can understand. And so I think that Liz and Jen did a great job with that. But this is the time to have questions. If you need further clarification on what CACNA1A is, what CAV 2.1 is, what types of mutations there are, please ask away. There is no silly question. I'm still asking questions all the time, so.
I was going to just ask to break the ice. Liz, if you could comment on, is there any understanding about whether a certain type of mutation, missense, frame shift, nonsense, et cetera, is tied to any particular severity? Like, do we know are frame shift mutations more or less severe than missense, et cetera? Do we know that for CACNA1A specifically, or any other neurodevelopmental gene?
Is this on? Yeah, okay. So to be clear, before I answer this, I'm going to say I am not specifically a CACNA1A expert, and so there may be other people here who want to weigh in on this question as we move forward. Generally, in ion channel genes, what we actually see is that the gain of function variants can often be associated with the earlier onset and more severe presentations compared to loss of function variants, which tend to often be later onset. In the slide, we sort of talked about loss of function, less likely to have seizures. The seizures may be later onset if they do occur, compared to gain of function, where you often are more likely to see status epilepticus, epileptic developmental and epileptic encephalopathy, and those sort of more severe earlier onset presentations. So as a general rule, I think actually the missense variants associated with gain of function are the more severe, and the loss of function are less severe. But I don't think that we know enough. We need more functional data, and we need more natural history data to be able to tie this together better in order to be able to make a prediction. I think usually we're coming to this information retrospectively, right? So we know the clinical presentation that the child has, or we sort of have an idea, and then we're doing genetic testing and finding the variant. What we aren't good at right now is doing testing and being able to make a prediction about what that might look like moving forward. And that will hopefully come with time and more information. But I know that piece specifically can be really frustrating for families, especially if you have maybe an infant. We can't always say exactly what things are going to look like in the future.
I wanted to add to Liz's comment. So about the, I couldn't comment on the severity, but what we did analysis is that comparing all the missense variants, their disease burden versus those that has protein truncating variants. And for the CACNA1A specifically, we nearly don't see any signal for the truncating variants in the DEE, but we see a huge signal of the missense variants. Now, this missense variants include both loss of function and gain function in this analysis. So suggesting that at least the missense variants disease burden presented a lot more than just purely not having the other copy of the protein.
Okay, thank you, sir. There's a question in the back, and then we'll go to the front here.
Thanks for those talks. Really nice explanations of the science. Damien Bell, Sophian Bioscience, I should add it as well. Following on from Sunita's question around severity, and somewhat what I guess Jen Pan touched upon at the end there. When you are doing these molecular characterizations of the variants of unknown significance, I guess because there's so many of them, you're almost trying to sort of somewhat predict what a certain mutation may be doing to the channel sort of properties and how it might be performing physiologically and pharmacologically within the body. Is that almost what your work will be doing, Jen, as in you'll be sort of trying to take as many of these variants of unknown significance and potentially grouping them based on their activities within the body?
Yeah, no, it's very much so. I think there are hundreds of the variants of VUSs that has not been dissected functionally. So what we're doing here is about 30, 40 at a batch, which is not quite the speed at which we wanted to go through all of them. So certainly, hopefully, the prediction algorithm is catching up with some of this pathogenic versus non-pathogenic. I wanted to say that, you know, we did try the alpha missense, which developed by the AlphaFold team. So in our in the alpha missense prediction for the benign variants, they got 1 out of 8 run, and then for the de novo variants, they got 2 out of 41 run. So not bad, but not great, because you know you want it to be a little more precise, and particularly for the CACNA1A, because the directionality matters. And then this alpha missense could not predict gain of function versus a loss of function, so that is a big gap. I think, you know, we could take two approaches. One is to do the brute force and then just buckle down and then perform all of the 350 variants that's known so far. It has been done for other channels, but has never been done for this class of proteins. And so that's one approach. Another approach is taking a saturation mutagenesis approach. It's using the calcium imaging as a readout, and that can be done as well. But both of them needs a lot of experimental try. Yeah.
All right, I think we've got time for one more question right now. There's right in the front. Go ahead and ask your question.
Hi, I was curious, you spoke to Jen, correct? You spoke to domains 1, 2, and 3, but not 4. Is there a reason for that? Because I know specifically our variant lands in domain 4.
Okay, it's a very good question, the sharp eyes. So the reason we did not include the domain 4 variants there is in our analysis, only one of the domain 4 variants made it to the second stage. So most of the domain 4 in our assay did not express much at all. So they basically, in our interpretation, they're loss of function, except one of them, which tends to be the other direction. So that's why I didn't put it there, because it's just one, there will just be one.
Can you speak to which variant that is?
I can show you after.
Okay.
Yeah, I can show you exactly.
Okay, last question from Lisa, and then we're going to move on to the next session.
Yeah. Sorry, I'm listening to this, and I'm just wondering if people might have this question, which is, if you test a VUS and you find that it's a loss of function or a gain of function, what happens to that VUS? How does it get from a VUS to being classified as pathogenic or likely pathogenic? Because parents keep hearing, well, your variant's a VUS, so we don't know anything about it. But if you're doing the functional characterizations, which we know we need desperately, how does can you just explain to people how that process?
Sure. Yeah, so we think of variant classification as being dynamic, right? This is something where we're always getting more information. And so if you have a result that's a VUS, I would just say don't give up. There is definitely a possibility that that variant could be reclassified. So most laboratories have a process where they proactively do some assessment of their variants, and they will proactively issue updated reports if they know that there's new information about a variant that might change the classification. But that process relies on, kind of doing this big assessment. Most labs do hundreds or thousands of tests in a week, and so you can imagine that process can be a little bit cumbersome, and even if it's automated, sometimes things can be missed. So what I always encourage families, if you have a variant of uncertain significance, once a year, check in with your doctor and ask them to contact the laboratory. Laboratories will do a reassessment of that variant for you and do a more comprehensive look. So it's kind of an individualized opportunity for us to take a look at your variant and see if there's any new information. Generally, laboratories will look for information that is published. So, after researchers complete these studies, they will publish them in the scientific literature, and that's what gets used to do reclassification. It varies from laboratory to laboratory whether they'll use unpublished data. If you know that you participated in a research study and there was some functional studies that were done that showed an impact and you have that information from the researcher, sometimes the lab and the researcher can connect and share and be able to do a reclassification based on unpublished data. So it's something to ask about. I can't promise if it's unpublished that will work. I know our laboratory will consider those, but I don't know that all of them do. But I think just be proactive in asking and have your clinician who ordered your test follow up with the laboratory to check in.
When are you planning on publishing, Jen?
It's already submitted. So Dennis Lau has a CACNA1A variant portal. If you apply, you could have access to these data as a researcher, I think. Yeah, so.
And the portal's fantastic because it's publicly available. So that's really great for giving laboratories a place to be able to reference, even if data is unpublished, but it's in the portal. So just wanted to call that out. It's a really good resource.
All right, so again, let's thank Jen and let's thank Liz for this wonderful first session. Thank you, you guys.